This is a video that I'm submitting to AHEC, just giving an overview of what I do at Chandrasekhar Community College as science faculty. So my name is Brent Voels. I have a doctorate degree in biochemistry and molecular biology, and I've been teaching at Chandrasekhar Community College for 10 years now. This is year number 10 for me. And the main thing that I really enjoy doing with students is all the different activities that I can offer to them that include research opportunities, many hands-on experiences, and things that can really propel them forward in their future careers. So I'm starting out the video just looking around at the research laboratory in the background. And so this is where my current two research students that started working with me in the summer of 2023 are operating out of. And you probably have some videos from them that are detailing the different techniques that they undergo. And so this is kind of one of the labs that we work out of, that we do the DNA extractions in here, run the agarose gels, and then visualize the gels, and all that happens in here. Currently, my two research students are actually working on 3 individual projects when you actually evaluate what's going on there. So right now, they're working on 3D printing weather stations. So they've been very much so working hard at that. It's quite a bit of troubleshooting. If you've ever worked with 3D printers before, they seem to always be breaking or just acting a little funny. And then the other project that we're working on, as you can probably see from all these little cup containers, is they were going out and catching pollinators during the summer months. And after we capture and catalog the pollinators, we then take them and they undergo a dissection of their gut microbiome using this equipment here. You can see the tools that they're using. And we are extracting their gut microbiome and then using a column kit to extract all of the DNA from that. We then utilize different primers to confirm the species of the insect that was captured. And then we use a set of other primers to analyze that pollinator's entire microbiome, including what type of pollen they might have been consuming. And this starts to overlap with the weather station project in a sense too, because the analysis of the pollinator's microbiomes is going to give us a fingerprint of what the pollinators in this area are like now. And then in the future, as these studies continue, hopefully at least other scientists carry this on, even if I'm unable to, we will see how their microbiomes might be changing in response to changes in the climate. We also have that third project that the students work on where they're going out and capturing mosquitoes, so we can verify and analyze their relative abundance of each species during each month. And that'll also play a role linking back to climate variables as well once we get those weather stations running, because it'll allow us to use the weather station data to pair with changes in either pollinator prevalence or mosquito prevalence. And a little interesting tidbit if you didn't know this, but mosquitoes are actually pollinators too. Mosquitoes actually only feed on a blood meal when they need to lay eggs. Other than that, they function as pollinators in the ecosystem. And this type of research really links back to overall human health, because our health is really dependent on having a healthy environment for which humans can live and exist in, and that requires a certain amount of biodiversity. We know that a huge percentage of our crops are actually dependent on pollinators, so it's really important that we understand what's going on with our pollinator populations as climate change starts to impact them. And that kind of wraps up just the overview of the type of research that I'm doing here. I love working with research students and teaching them all these new techniques and giving them plenty of hands-on experience, and hopefully it propels them forward whatever direction they choose to take their career. So this video I'm adding in as an overview of the science program here at Chandeska-Cheeskana. My name is Dr. Brent Voels, and I've been science faculty here now for 10 years, and I teach the biology classes and the chemistry classes to our STEM students. And this lab right here is our main research laboratory. This has never been a teaching classroom. It is set up just to do at the bench work, and showing off some of the different equipment that we have here. So I have this PCR chamber here. This is really important. It allows students to get good results and prevent contamination of their DNA samples. So we're going to be utilizing this, and so students are going to learn about the importance of aseptic technique and keeping your samples clean. It's really difficult to do PCR because there's actually so many floating particles in the air as is that if you really want to make sure your results are good, as good as possible, you'll need to use something along these lines. We also have access to a digital PCR with an automated droplet generator. So this allows us to do really, really precise quantification of specific genes that might be present or absent in a sample given certain circumstances. This is a very advanced technology, and the ability to do digital PCR sets students up where in this setting, in this research setting, they're capable of doing every kind of PCR, from your conventional PCR to your real-time PCRs and now digital droplet PCRs, so they can get cross experience on all those different platforms. They all have their own unique uses. Like currently in our lab, we're mostly doing conventional PCR because in the case where we're doing sequencing, you just do conventional PCR because it's the most cost-effective method, and these other ones aren't really as necessary. We have different centrifuges and things set up depending on what's going on here. Here is a really nice spectroscopy reader that reads on 96 well plates. This one's a really nice one. It does luminescence and absorbance and fluorescence all together. Instead of your traditional hot water baths, which are actually prone to contamination and growth and can lead to all sorts of issues, I've opted for a stainless steel bead bath, which is really nice. It's technically just a lot cleaner and better for the lab, easier to set up. You do have to make sure it gets warm to a certain extent before use. I have a lamellar flow hood for when we need to do different things that require a more sterile environment. Again, going back to aseptic technique. Students could also do their PCR work in this hood if the other one's used and a second student needs to utilize this one. We have our real-time CFX 96 well plate reader here, so this can do the real-time PCRs. And then we also have the trans turbo blot system. So for Western blotting, this can cut down on the amount of time the students need to spend doing that technique. Then we have our conventional thermocycler, which is actually a pretty big workhorse here in the lab. So those are some of the different equipment that's available for the students who are doing research where molecular biology techniques are going to be heavily used.
Right?
Hmm?
The ladder is different.
Yeah, good observation. So you'll probably just want to do another dot for the ladder, because the ladder, remember, is 5 microliters dye, 2 microliters ladder. So I have a feeling you might have done.
I must have went too high.
That's okay. It's the beauty of the parafilm dot method.
So today I'll be making a gel for electrophoresis. And I start by getting 0.5 of agarose powder. So as you can see, I've already put the powder inside of the boat, and we have about 0.5 grams of agarose powder. This is a running buffer for the electrophoresis process. It was mixed using 100 TBE buffer, and we filled the rest with double distilled water up to 500 milliliters. Now we'll be confirming that it's mixed by swirling it, making sure that there is no buffer present, and it looks pretty clear. And now we'll be taking our agarose gel and adding it to this flask to make the gel. And now we'll be adding up to 100 milliliters. And I got that. Now that we have our agarose powder and running buffer here, we're going to use a microwave to help dissolve the agarose powder better. Using 30 seconds for the first boil, and you gotta watch it very carefully so it doesn't boil over. Now that our agarose powder and running buffer have boiled, we're just going to swirl it to try to get it to mix a little better. And then we will put it back in the microwave for another second boil. Okay, now that the second boil is done and the agarose powder is completely dissolved, we can move on to the next step. I'll be adding 2 microliters of ethidium bromide into our gel mix. Swirl to mix the ethidium bromide, and now we'll be making the gel itself. Okay. Now we'll be placing in the fridge where it will solidify within 20 minutes. Okay.